So why don't we start with a sound check. Can you hear me in the back? Yes? Can you hear me in the back? Wonderful. Thanks very much. A question was raised on Monday about aspects of initiation in eukaryotes and so forth. So I just want to clarify some things and be sure we're all on the same page together. It is the case that for both prokaryotes and eukaryotes, initiation always starts with methionine. It's also the case that it always uses an initiator tRNA that's specific. Now, in prokaryotes, that's an F-met tRNA. In eukaryotes, it's just an initiator tRNA, but they're specific ones. In prokaryotes, as I mentioned initially, there is a sequence that is called, oh, I'm sorry. Oh, okay, let's see if this does, oops. All right, let's try, interesting, okay. There we go. All right, thank you. I can't see, contrary to what my children believe, I do not have eyes in the back of my head, and so I appreciate being warned about these things. And so, let me get that arrow on. Okay, so the Shine-Delgarno sequence on the prokaryotic messages allows there to be an interaction between the message and the ribosome before the AUG. That is, Shine-Delgarno is what we call upstream. And it's very important that it works this way because, if you remember, many prokaryotic messages are polycistronic. They encode multiple proteins. And so having an approach like the Shine-Delgarno sequence means that prokaryotic messages can have ribosomes come in and land on them in several different places to make proteins. So it allows for something called internal entry. So you can land in many places. So that's the prokaryotic approach. In eukaryotes, as for so many things, you can make mostly statements, right? So about 90% of the time, the first AUG will be used on a eukaryotic message. And about 90% of the genes, this is you don't have this. This is an addendum. It'll be on icon, okay? I don't have a handout for you. Just look at the screen and think. It will be available. It'll be off of icon probably by the end of class or a little after. About 90% of the genes for abundant mRNAs use the first AUG as the place to start translation. Less common mRNAs, rare mRNAs, may have other kinds of control going on. So they may use an upstream AUG, an AUG that lies between the methyl GCAP and the AUG that's used for translation for the main protein. In some cases, there's an entire open reading frame upstream of the main show. There may be secondary structure up there that the ribosome has to get through and the helicase activity has to unwind. There's probably not something called an IRES, which is long shorthand for internal ribosome entry site. This is the idea of a ribosome coming in the middle like they do in prokaryotes, and this is the thing that I was a little confused about on Monday. I have citations here for both of these, so for people who want to read in more detail, and those articles are also available on icon for anyone who wants to read the whole presentation. So the way it works is that the 40S subunit will use ATP to scan along mRNA. You have to assemble all that stuff at the beginning, and the 40S subunit will scan along it. And 90% of the time, the first AUG is the one that's used. That AUG is in the context of the sequence that was described on your handout. This whole sequence is what's called the Kozak sequence, and it's not perfectly present around every AUG. The more closely it matches, the more efficiently translated the message is. So this is not an upstream message. This is a sequence. This is one that flanks the AUG. So in the prokaryotes, it's upstream. You get internal entry and can translate away. In eukaryotes, you're going to scan along, and whether or not you use that AUG will principally be determined by the strength of that Kozak sequence or consensus sequence, and in a few rare cases by other things. It's interesting to note that the messages on which things are more complicated than this are developmental messages, cell cycle messages, things that need to be regulated either very rapidly or independently of transcription. So these are messages where an additional level of control has been desirable. And if you're interested in those additional controls, you're welcome to look at those articles. And as I said, this will be available on ICON as well. Any questions about that at this point? Let's see. Which one? Okay. Eukaryotes use the first AUG. Oh, you're right. Well, okay, so I'm human. You're right, that should be AUG. So, just goes to show no matter how hard you prove things, you can still manage to miss them. All right. Thank you for catching that. We'll just save that. I'll replace the one that's on the web with this one then. Were there any other details that people noticed in here that they want clarified, expanded on, whatever? I know, that's another meaning of it, yeah. So IRES can mean several different things. This is the internal ribosome entry site, is what that one stands for. And so an internal ribosome entry site is the idea that a eukaryotic message would behave like a prokaryotic message and have a ribosome descend in the middle, bind, and go. Okay. There were some there were some people who thought they had found these things, and Kozak has this review is actually by Kozak, and she reviews the evidence for it, and she's not very persuaded. It's not something I'm right on top of. So if you like to watch slugfests, I would recommend it to you, and you can watch them duke it out. But from what I read of her comments, it seemed to me that people had not made a strong case. But I'd be happy to be corrected if people have more time to pursue it. Other questions? Okay, so I will put the corrected one up. Thank you for finding that typo. And we can go back to proteins. Apart from the initiation questions, are there any questions from Monday's lecture? Okay. So this is just a chance to sort of review the whole elongation cycle and have it fresh in our minds. That in order to add the next tRNA or the next amino acid, tRNAs have to be delivered to the ribosome through the effect or through the agency of an elongation factor, EF-TU, which, like so many other things in protein synthesis, has a GTP bound to it. Its delivery to the ribosome into the A site here is accompanied by hydrolysis of that GTP. So now you have a fully loaded ribosome. You're going to want to make a new peptide bond, and that's the activity called transpeptidation. So the growing peptide chain is transferred from the tRNA that's in the P site over to the A site. And that transpeptidation reaction is catalyzed by RNA on the ribosome, but does not require any input of energy at that spot. At this point, you need to move things along, and so that process is called translocation. So the loaded tRNA is shifted over to the P site. The messenger RNA is shifted over one codon, and the empty tRNA goes into the E site for ultimate removal. And EFG is going to use GTP hydrolysis to facilitate the removal of the empty to facilitate the removal of the empty tRNA. And here you see it having hydrolyzed and coming off. So it's going to cost you 2 GTP per cycle of elongation, in addition to the 2 high energy bonds it took to charge a tRNA back at the beginning. So, or 4 high energy bonds for each peptide bond. Okay. So you initiate, we talked about that. You elongate, you've got the cycle going around and around. Even the longest message at some point comes to the end. Titan protein is 1 million molecular weight, and yet at some point even that message is done. And so how does that happen? So here we have a ribosome that's gone through multiple rounds of elongation, and the message has slid over, and the loaded tRNA has slid over, and it's sitting so that the A site is now occupied by a stop codon. There are 3 stop codons. Don't expect you to memorize the stop codons, but there are 3 stop codons that just mean stop. This is the end. At this point, a release factor has to come in and enter and sit in that A site. The release factor can then catalyze the transfer of this peptide chain onto water instead of onto a tRNA. And of course, if you transfer it onto water, it's now free. And so by catalyzing its transfer to water, you now have a ribosome that has a tRNA in the P site, you've got a release factor in the A site, and a free polypeptide. This by itself might well be sort of locked in place, and except for the recovery of another release factor coming in, which can catalyze the removal of the the first release factor, so that that can dissociate. And then there are other factors that take part to open the ribosome back up into 2 pieces so that it's ready to enter the cycle, enter the pool of ribosomal subunits that are needed. In eukaryotes, there is just ERF1, and it has a GTPase activity in the molecule, but there's not a need for anything else to do it. Release factors have structure at their ends that look like an anticodon, enough like an anticodon to bind at that spot. And you can actually map, where did my, I know where my pointer went. You can actually detect what in effect is an anticodon, a 3-amino acid sequence that is the mimic, the molecular mimic for that anticodon. Okay. I talked about being able to restore these to the pool. In prokaryotes, there's a ribosome recycling factor that cooperates with EFG, which is normally in the elongation cycle. The ribosomal release factor can bind at that A site. EFG can move it over. That bumps out the other tRNA. And then at that point, it can fall apart. And this is the structure of the ribosomal release factor. This is just a reminder one more time of the exam on Friday and the final exam on May 7th. So, we've talked about so far messages and ribosomes as a one-to-one matching, as if this message has a ribosome associated with it. But a messenger RNA is a very long thing by comparison to the part of it that's occupied by a ribosome at any one time. And so once the ribosome has moved down the message far enough to expose the initiation site, it is possible for another ribosome to bind and start translating. And this is an example of what that can end up looking like. This is what's called a polysome, which is short for polyribosome. And if you look at this thing, you can see that the ribosomes have squiggly stuff coming off, and the squiggly stuff is getting longer the further down the message you go. So here's one messenger RNA that has, I don't know, 15, 20 ribosomes on it. And if you look in tissues with large proteins, like muscle, for instance, myosin is 200 kilodaltons, you can see polyribosomes in the tissue that are the right size for something translating a protein that big. There's a lot of energy invested in translation, so it would be good if you didn't start it if it wasn't a good message. That is, quality control would help, not just quality control at the level of particular residues, but quality control at the level of the whole message. I mentioned earlier that the poly-A binding protein is in effect part of the initiation complex, and since the poly-A tail is at the 3' end, what that means is that in eukaryotic messages, a fully intact message is actually a ring-like structure, in which the 5' methyl G cap assures an intact 5' end, and the poly-A binding protein assures an intact 3' end. And so in addition to providing information to start here, it's also providing information to say that this is an intact messenger RNA. That's in eukaryotes. Now, prokaryotes don't have poly-A tails. And so prokaryotes can't use poly-A binding protein's presence or absence as a clue to the ribosome that this is worth putting your effort into. What kind of control over quality is there possible in prokaryotes? And it turns out to be done in kind of a cute way. So prokaryotes have a 10S RNA, or a small stable RNA, that has the properties of having something that looks like, in effect, a tRNA, and something that has a little bit of message. And so the idea here is that if you have a ribosome in which you have a tRNA that's loaded with a peptide chain just sort of stuck there because it's a defective message, there isn't any more down there to go, this small stable RNA can come in. And can be charged with alanine. So this is an RNA that can have a covalent bond to an alanine. And it binds in the normal place for the messenger RNA. It triggers the ribosome to transfer the growing peptide chain to that alanine. And then this message would be released and could be degraded because it's no longer being translated. Being translated is a protection in prokaryotic messages. The little bit of the ssRNA that acts as a open reading frame then acts as the template for several rounds of additional peptide bond formation. It has a fixed sequence in there, so when it's complete and the ribosomes are released, the protein that was translated that way has a tag on it that says this came from a bad messenger RNA. And so that protein is now tagged to be degraded because it came from a defective message. And the usual kind of release will work in order to recycle the ribosomes. So it's a single RNA that can function both as a tRNA and as a messenger RNA. And it provides quality control. And this is just showing you those pictures in a little more detail. Here's your stalled defective message with the tRNA in the P site and the growing chain. One of the reasons I picked this is it's one of the few pictures that shows you the E site. Not very many of the diagrams show you the E site. And so here it's stuck. You can't get a release factor in because there's no message there to bind it. In comes the ssRNA. The tRNA looking like part can trick the ribosome to making that transpeptidation reaction that's catalyzed by RNA. And then that stimulates the movement of the initial tRNA over into the E site. The mRNA part of the ssRNA is now going to act as a template for translation. And then when you're all done, you have this peptide that has the tag on it that says this is not good. And the sequence, the open reading frame on the ssRNA includes a proper stop codon. And because it does, you can get a proper release factor in there and use the normal machinery for opening up the ribosome. Any questions about that? Okay. Okay. Just as RNA often needed some kind of processing to be fully functional as RNA, protein also has to undergo folding at least, and sometimes processing. And so there has to be an environment or the context or the machinery to let that happen. And if we were to look at the effects of correct and incorrect folding, this is sort of a schematic for that. You have here your newly translated polypeptide. If it starts to fold up correctly and the hydrophobic domain that should be at its center gets folded or the molten globule it's sometimes called, is there at the center. The hydrophilic parts of the peptide can now wrap around that hydrophobic core and make a nice native molecule. If something goes wrong, if this hydrophobic domain is not correctly aligned for the rest of the things to align around it, you can end up with things like this happening from either of those sides, where you have several copies of the protein tangled together through their hydrophobic domains in an aggregate that's not functional because it's all just held together. We used to think that protein folding was completely spontaneous, that all that needed to happen was that the linear sequence was there and it would fold up. This despite the fact that protein chemists often couldn't refold proteins. The more we know about cells, the more tricky it seems that proteins can fold successfully. And one of the things that was often not taken into account in thinking about protein folding is how crowded the inside of a cell is. So it's quite common for the interior of a cell to have 30 to 40% of its volume occupied by protein. And so as peptide chains are being translated, long before they actually are completed, they're going to be bumping into other things. And so perhaps we shouldn't have been so surprised that cells have proteins that help proteins fold. These are called chaperones. They're called that in the sense that they want to prevent inappropriate associations. You know, chaperone used to sit between the dates to keep them from interacting. And so the idea is that the chaperones will keep these proteins, you know, isolated long enough to fold up and make a good protein. One common way that they work is to cover the hydrophobic regions to keep them from being stuck to by other things. Some of the chaperones actually can use ATP to induce conformational changes that allow some unfolding or to release the folding. This is an example of a heat shock protein, and these are named for the fact that although they're present at some level, most of the time, they are induced in cells that are subjected to heat shock and in some cases other shocks. Because during a heat shock or some other stresses, the environment is one that tends to make proteins denature. And so the heat shock proteins have a role at trying to keep that to a minimum so that the proteins are as functional as possible. This is an example of a multimeric chaperonin where there's actually a chamber in the inside that allows all this process to happen. I think we have a picture, on how that actually works. And this is a... we had no idea that it took this to make a good protein, okay? So here you have an open, empty chaperonin, if you like. It has to bind ATP. Once it has bound 7 molecules of ATP and an unfolded polypeptide, so here's your unfolded polypeptide. There are hydrophobic patches in here that this can interact with. And the combination of the presence of the unfolded protein and the ATP facilitates the binding of a cap, if you like, called BroES. And so it sits down over that opening, and that changes the interior of that chamber. It sort of tucks away the hydrophobic patches so they're not there anymore. And it gives it a space in which it can fold without interacting with other things. After a brief period, it's long compared to a protein, but brief to us, 10 to 13 seconds, the ring of GroES have hydrolyzed their ATP. And that then causes the cap to release, and in concert with binding at the other side, you have release of the folded protein from the top while you're sort of filling up the bottom chamber at the same time. The idea this is trying to convey is it's an either or, that it works at one end and then the other back and forth. It's not, both sides aren't working per time. So it costs 7 ATP per event, per time that a peptide goes in there. It's a fairly expensive process that way. And a surprisingly large fraction of the cellular proteins need this kind of help, up to, a seventh to a fifth of the cellular proteins will not fold properly without the assistance of chaperonin. Some of the other kinds of structural enzymes, if you like, or conformational enzymes, include proline isomerase. You remember that proline is the funny amino acid that has a ring that closes back around, so that the side chain comes back around and closes with the amino acid part. And those prolines can be either an a cis or a trans configuration in a protein. And flipping between those two configurations can happen spontaneously, but it's not terrifically fast. The enzyme proline isomerase can catalyze those conversions. Another enzyme called protein disulfide isomerase is concerned with cysteine-cysteine sulfhydryl bonds. So if you have a disulfide bond, where the two sulfurs are bonded together, if the protein only has two cysteines, it can only make one disulfide bond. If the protein has 12 cysteines, it can make a whole bunch of disulfide bonds, and I don't want to go into the math of it. And one of the jobs of protein disulfide isomerase is to break that disulfide bond and try a different one to see if that one works better. Now, we've been talking so far about things happening in the cytoplasm, proteins in the cytoplasm, but of course, that's not the only place that proteins have to be. Some proteins have to be in membranes, and some proteins need to be on the opposite side of membranes. So for instance, our antibodies need to be outside the cell. Hormones, peptide hormones need to be in a vesicle that can be released and so forth. And the way that happens is a little different between prokaryotes and eukaryotes. In prokaryotes, the protein is made and folded, and then the folded protein is transferred across the membrane by specific machinery. And that kind of process works in eukaryotic organelles as well. As you may remember, eukaryotic organelles have some of their own DNA. They depend on the nucleus for a lot of it. And so those proteins that are translated in the cytoplasm have to get into the mitochondrion or into the chloroplast so that they have the machinery they need. For eukaryotic proteins, the ones that are coded through the nucleus and are meant for the cytoplasm or the extracellular space, that kind of transit happens co-translationally, and it looks something like this. It's going to depend on a signal sequence, something that has an overall features that are favorable to this, and it's going to depend on a signal recognition particle, SRP. And here's a picture of SRP. And SRP is both protein and RNA. It came as quite a surprise to people that it took RNA to facilitate the transfer of proteins through membranes because it's such an important process. The signal sequence is not a very precise sequence. It's 6 to 15 residues that can form an alpha helix that's fairly hydrophobic. And then after that sort of nebbishy sequence, you have a bunch of histidines. The hydrophobic part can stick to or reside on the methionine of SRP, and the RNA part will interact with the histidines to neutralize their charge. And part of what that does is that if there's a signal sequence coming out, it encounters SRP, SRP gloms onto it and arrests translation. So that message will be partially translated. SRP snaffles onto the signal sequence, and nothing more is going to happen until the next appropriate things. And this is what needs to happen next. Where are we here? So we've got the situation of an arrested message. So we've got this signal peptide. SRP has, let me see here, bound to it to arrest it, it provides a transfer to specific places on the endoplasmic reticulum. So there are receptors, if you like, for that complex. And what those receptors permit then is for that signal sequence to go through that membrane and release the inhibition on the ribosome from translation. So once this ribosome is docked appropriately on the endoplasmic reticulum, translation can continue. So you can see the messages being reeled through and more protein is being made. And in most cases, the signal sequence will be clipped off, not every case, but most cases, by an enzyme that can specifically recognize that. And now you have a message that's translocated into the ER lumen, or depending if it's a transmembrane protein, part of it has stayed in the membrane. There's a lot of interesting machinery that allows for sorting so that the right proteins get to the right vesicles and so on. Okay. In addition to needing to fold up, a lot of proteins need to be modified after translation as well. I know one protein chemist who claims that if you look closely enough, all proteins are modified. I'm not sure I quite believe him, but I do believe that the extent and variety of modifications continues to grow the more closely we look at things. One kind of modification we've already seen is proteolysis. You may remember that in the case of chymotrypsin, it's made as a precursor, chymotrypsinogen, that has to get cut through proteolytic processing. Another example is this one, which is insulin. Insulin is synthesized as a single long peptide. It folds up to have the correct disulfide bonds here between the A chain and the B chain, but there's a cleavage that separates this intervening bit of peptide called the C chain that then comes out and is released and degraded. Interestingly, the C chain of insulin is probably the most rapidly evolving sequence that we know of, because the only feature it has to have is to be long enough it can tolerate almost any kind of substitution. Another very important modification is glycosylation, the adding of sugar groups. Sometimes this is as simple as just one sugar. Sometimes it's an entire little palm tree of multiple oligosaccharides that are themselves modified, depending on the purpose and the function. Most glycosylated proteins are going to be heading outside the cell. Not every single one, but most of them. Other kinds of modifications include various kinds of hydrocarbons in effect. So you can add a methyl group, an acetyl group, a palmitoyl group. So a single methyl group is not very big. It can often block a charged residue, so it's now not charged. A palmitoyl group, of course, is longer. Palmitic acid is 16 amino acids. That's long enough to actually anchor it into a membrane or something. The amino terminus is often acetylated, and that has the effect of neutralizing the charge there and permitting folding in different ways. The C terminus is often amidated, so the ends of the molecule are often modified. Okay. I've tried to emphasize that whenever there's an on switch, there's an off switch, which is almost always true. And so, in addition to protein synthesis, there will be protein degradation, and there will be some way to control protein degradation. And that is true. And the largest volume of intracellular degradation is handled through proteasomes in eukaryotes. Looks kind of like a barrel. It's a multi-protein subunit. And the way in which you get sent into this barrel to be eaten up is to be tagged with ubiquitin. Ubiquitin is a small, extremely conserved protein whose principal function is to target things this way. That puts sort of the final question back one more: which ones get ubiquitinated? In many cases, we can see on short-lived proteins something called a PEST sequence, which is hydrophilic stretches, at least 11 amino acids long, that have at least one proline, one a glutamate or aspartate, one serine or threonine, and then either a lysine, an arginine, or a histidine for some positive charge, and then no positive charges inside it. So there's a kind of a general consensus-y thing that apparently is a good flag to get these things put onto it. Okay. All right. So the rest of what I'll be talking about is actually, let me just stop and ask, do people have questions about any of that material? Okay. The rest of what I'd like to talk about is a little bit about some work that was done in my own laboratory as a way to give you a little bit of a flavor for how some of these things connect in specific situations and the way that they can be used by the cell to do interesting and creative things. I don't know if we'll have a chance to get through the stuff, but it will let you see connections between a number of the topics we've talked about along the way. So polyribosomes, like this, we just talked about those today. They're a messenger RNA that have more than one ribosome on them translating. In eukaryotic cells, the majority of polyribosomes that aren't on the ER are on the cytoskeleton, which is a matter of some interest, that they are not just floating free in space, but that they're anchored to the cytoskeleton. Now, what's of interest is that nascent proteins that are still growing out of a ribosome, can, in some cases, attach to the cytoskeleton. And so, in this case, instead of seeing co-translational insertion into the ER, we're seeing co-translational assembly of some cytoskeletal proteins. And how would something like that work, and what difference would it make? Well, let's see now. How can we know whether they're doing it? Well, one way of seeing nascent chains, incomplete chains that are in the process of being synthesized, is to give a little bit, a short period of time of radioactive methionine, and then chase that with cold methionine. And so the radioactive methionine will label specifically the proteins that are being translated at that moment. You can then use antibodies and pull that protein selectively out of solution, and then use gels to separate it and show the sizes of those proteins. Since the proteins are radioactive, you can just simply put them on X-ray film and they'll take a picture of themselves, in effect. So that's one method of doing it. And there should be a picture in there, and I don't know where it's... Well, the picture is in your handout. I don't know why it's not showing up on the screen here. You can see in the picture in your handout the presence of peptide chains that are less than full size that in fact become full size if you allow for further time for synthesis. So is there a pattern to which proteins do this? Well, let's take a little look at this. Myosin heavy chain we talked about earlier. It has a coiled tail. About 30% of myosin heavy chains associated with the skeleton this way. Titin is another muscle protein. It's huge. It's 1 million molecular weight. It spans half the sarcomere. Greater than 80% of it assembles during translation. But those are both muscle proteins. Vimentin is an intermediate filament protein. You may remember them. They're the coiled coils that are not polar. And it depended on which cell you were looking at. So less than 10% of that protein assembled during translation in a red blood cell, but about half of it did in a chick fibroblast. In NDCK cells, this is a kidney cell. In kidney cells, how much of it assembled that way depended where in the cell cycle. So we talked during the cell cycle part about a variety of things that change during the cell cycle. This is another aspect of it, of how the cell actually assembles its machinery. Oh, there's the picture of it. Okay, so here you can see the picture of, at the end of a pulse, there are less than full-size chains, and after giving more time to complete it, they all end up at the full size of myosin heavy chain. Okay. Now, maybe cytoskeletal proteins do this because the cytoskeleton is a sticky thing, or it's an insoluble thing. But that's not enough of an explanation, because tubulin, which is an authentic cytoskeletal protein, it makes microtubules, which are polar and the big sort of robust things that vesicles can move on. We could not detect any co-translational assembly from them. And in fact, other groups have shown that tubulin seems to need chaperones to fully fold. Alpha-actinin is another cytoskeletal protein. It's a protein that helps connect actin to things. It again did not assemble during translation. Non-muscle actin, in our hands, less than 10%. It might be 0, but it's certainly not more than 10%. So if some proteins do and some proteins don't, the next logical question is to ask, can you predict which ones do and which ones don't, and why perhaps they do or don't do that? Well, the proteins that do assemble to some extent by co-translational assembly include vimentin, which is a parallel coiled coil, myosin, which has a parallel coiled coil tail, titin, which is a really long, skinny protein. It sits on long, skinny structures. I didn't mention this, but tropomyosin is another one in here. Proteins that don't assemble co-translationally include tubulin, which is a globular dimer, alpha-actinin, which is an anti-parallel coiled coil, and oops, let's go back, and actin, which is a globular one. And so if you look at these features, the ones that are doing it are either parallel coiled coils or long skinny proteins. The ones that aren't doing it are globular, anti-parallel. You couldn't do that co-translationally, could you? So if we use those features, we can then identify a protein that might be a candidate, if you like. Tropomyosin is a parallel coiled coil that sits on long skinny structures. And so it has two of the features that some of the other proteins have. And in fact, when we measure the behavior of the tropomyosin in muscle cells, yes, it did. If we look at tropomyosin in non-muscle cells, no, it doesn't. Now, the tropomyosin in muscle cells is differentially spliced from the tropomyosin in the original transcript. So the muscle tropomyosins have an additional domain on them compared to the non-muscle tropomyosins. It's another example of tissue-specific protein expression. And in this case, differences in how they actually behave. So what? I mean, does it make any difference that some of the skeleton assembles that way? Well, for any protein that assembles during translation, its message is next to its site of assembly, its initial assembly. So that suggests that there might be a role for messenger RNA positioning. Now, once assembled, things could rearrange. They might go through more than one round of assembly. So co-translational assembly does not imply that all the messages are going to be localized with the protein, just that they may have a pattern of localization. And this is just a caveat that you might need to rearrange after initial assembly. And so I'd like to show you some localization of messages. Titin is this huge million molecular weight protein that is a structural protein. Vimentin is a non-muscle protein that's common to many cells. Again, a parallel thing. And so let's see where they go. Here is a picture of a muscle cell. This is a little bit of this muscle cell. If we stood it on end, its top would be well out the top of the building. Muscle cells get very long. And let's see. This is showing you in black the position of the messenger RNA, of the protein here in the top, an antibody that recognizes just one little bit of titin. There's titin protein clear throughout there because it's so long, but that's just one part of it. And this is showing you the messenger RNA. So you can see the red is where this part of the message is, and the green is where that part of the protein is. This probe recognized a different part of the molecule than that antibody, so the fact that they don't overlap is in fact consistent. And that's just showing you some more of that architecture. Here's an example of the Vimentin message. Here you have the protein shortly after a muscle cell has elongated to reach out and go through the fusion thing. Its Vimentin message is still largely near the nucleus, but the protein has rearranged in the last few hours. This is showing you that picture as a serial section to show you that it's not just the chance of that one section. That was the pattern of Vimentin message in a young muscle cell. If you allow muscle cells to continue to grow in culture until they start to twitch, you continue to develop architecture, the sarcomeres develop, the muscles, the myosin, the titin, the actin all get correctly positioned, and you can end up with the Vimentin being organized as well. And so you can see here the co-localization of the Vimentin message and protein, with the exception of some granules, and these granules are probably transport packages that have been studied in other cell types. So fibroblasts have very different architecture. This may be more similar to what you were used to seeing in our first part of the cytoskeletal discussions. So here you can see a Vimentin that's been tagged with a marker so we can distinguish it from the rest of the protein. These are, again, the cells at rest. They're not moving anywhere. The Vimentin message is to one side of the nucleus here. You can see it off to one side of the nucleus. That side is the same as the centrosome, which is where microtubules are organized. You can make those cells think they're in a wound. You can make them try to heal, and it looks like this. You can grow a confluent monolayer, scratch the disc so there's nothing in the middle. The cells here on the free edge believe that it's a wound and they try to move into it. And there's the messenger RNA. There's the centrosomes. And so what they do is they reorganize their message to the front side of the cell where it's going to move forward. And they rearrange that message as one of the first things they do when they orient to the wound. So we see a consistency between protein and message position. We see it change during development. We see it change with behavior. Is this all coincidental, or does it actually make a functional difference? And the way we can ask that is we can make a Vimentin that we can follow. So we've put on it an epitope that lets us see it. We change where it goes. Messenger RNAs have a 3' untranslated region that varies in length, has a number of different things happening in there. One of the things in there turns out to be something that you could call a zip code that allows it to be delivered to the correct address. And we can give Vimentin the zip code of actin. And if we do that and put it into fibroblasts, we can ask, does it change the shape? Does it change how it moves? Does it change cell division? Does it change anything? Does it make any difference at all? And so this is a fibroblast. Now you just saw a fibroblast a minute or two ago. This doesn't look like a fibroblast. The only thing that this fibroblast differs in is it's being forced to make its Vimentin where it normally would make actin. And so it has completely changed the architecture of the cell only by changing the position of the message. If I give this a Vimentin UTR, the cell looks perfectly normal. So this is after 3 days of expressing it. This is a sort of a time course of that. Just even more dramatic effects here of how mislocalized Vimentin can do it. This is again showing how the RNA is now out in these processes. This is not a muscle cell. This is a fibroblast in which the message is now out in the process. And we can look at movement. And in the case of moving cells, remember I showed you that the Vimentin message was put towards the front end of the cell where it's going to be spreading out. If we misorganize the Vimentin, they're left behind. Here's the edge of the wound, edge of the cells moving into the wound. The misorganized Vimentin cells are left behind and can't move. So that's the end of that story, and look forward to seeing you all Friday.